Hello, everyone. My name is Yusof. I'm from Leeds, and I'm reporting for Room Now at EULAR 2023, live from Milan, Italy. Today is day 3 of the Congress, and there have been so many interesting abstract and posters presented at the conference. And today, I'm delighted to be joined by Dr. Iaonnis Parodis from Karolinska Institute. Hi, Iaonnis.
Hello, Yus. Thanks for inviting me. It's my pleasure to be here with you.
It's a pleasure, for us as well. So today we would like to give a quick summary of Dr. Parodis' work. The poster is 1156, and today we will be discussing about de novo renal flares during Belimumab randomized control trials. So Iaonnis, would you like to just give us a brief summary, the background and the objectives of your study?
Yes, absolutely. So we designed this study because we had some signals from real-world experience with Belimumab. So some years ago, we published some cases of the Novo lupus nephritis during treatment with Belimumab. So these were patients that did not have renal involvement before they started treatment with Belimumab, and developed a biopsy-proven lupus nephritis during therapy. And since we felt that the cases were, let's say, more than what we could expect, we started investigating this a little bit more in depth. So we even matched the population of Belimumab-treated patients with the population of patients who had received other medications and were followed for quite some time. And we could say that patients who had received Belimumab developed the novel lupus nephritis to a greater extent. So this brought our attention a bit, and we thought, okay, maybe we should look into this in clinical trial data, to where we have a placebo arm. The patients are randomized, so there we have better, let's say, opportunities to look into protection or predisposition conferred from Belimumab with regard to development of de novo renal flare. So this is what we did, and we designed a post-hoc analysis of data from 5 phase 3 randomized clinical trials of Belimumab. These were the BLISS 52 trial, the BLISS 76 clinical trial, the BLISS Northeast Asia, the BLISS subcutaneous, and the EMBRACE, so Belimumab in black race patients. And the total number of patients who participated in these trials, this was more than 3,600 patients. And the patients who never had renal involvement until the baseline of the trials, this was 1,844 patients. And from these patients, it was 136 patients who actually developed de novo renal flare. And this was defined as a transition from BILAG E to BILAG A or B in the renal domain of the classic BILAG within the 52-week follow-up of the trial. So this was pretty much the design of the study and the rationale why we did this.
Okay, and I think you touched just now about some of your key findings. So you mentioned there are about just over 100 patients, so about 8% developed de novo renal flares. So in terms of predictors, do you see any predictors of this renal flare when you analyze it?
Yes, absolutely. So it's nice that you're bringing up the percentage, because it was actually pretty similar to what we reported a couple of years ago when we looked at real-world data coming from Sweden and from UK together, from Leeds, actually. So it was striking first to see that there was a clear separation between Belimumab, the intravenous Belimumab 1 milligram per kilogram, so the low dose intravenous Belimumab, and the placebo group, the respective placebo groups. So we actually did a crude analysis to begin with, taking the placebo groups from their respective trials in order to make sure that we had a representative placebo group for its comparison. So we could see a protective effect of the intravenous low-dose Belimumab against renal flares. We could not see this protective effect from the high-dose intravenous Belimumab. We could not see the same protective effect with the subcutaneous Belimumab. And this was the crude analysis. Then to take time to event into account, we did Cox regression analysis, first univariable and then multivariable Cox regression analysis, and there we could confirm that the low dose Belimumab was the Belimumab dose that gave the greatest protection against development of the Novo renal flares. In that analysis, actually, we even saw a protective effect conferred from the subcutaneous form of Belimumab, but there was no protection, no clear protection, at least, conferred from the high dose intravenous Belimumab. However, there was no predisposition either. And when it comes to other, let's say, factors associated with renal flares, it was a bit surprising to see that male sex was coupled with fewer events, so a protection, let's say, but the Asian population and the indigenous American population, these patients were more predisposed to develop de novo renal flares. Then anti-dsDNA positivity at baseline was a factor that was associated with development of renal flares. Then anti-cardiolipin antibodies, both the IgG and the IgA isotype, these were also shown to predispose. Then prednisone dose, increasing prednisone dose, so as a continuous variable, was also shown to predispose, and also the use of mycophenolate mofetil or oral cyclophosphamide. Some few patients in these trials used actually oral cyclophosphamide, and also the use of leflunomide. And I see these drugs and also the increasing dose of oral prednisone as proxies of disease severity, and on the other side of the coin, methotrexate, use of methotrexate was associated with a lower risk of renal flare development. So this was the summary, let's say, of the results.
Yeah, thank you so much. Brilliant summary. So in terms of implication to clinical practice per se, so in these patient with SLE with non-renal active disease, what is your advice, and what should we be looking out for, your recommendation?
So it is difficult to say. We could corroborate, let's say, the vulnerability of the Asian SLE population to renal affliction. With this study, we could clearly see a protective effect of the low-dose intravenous Belimumab against the development of the Novo renal flares. We could not see a clear protection. We could not see any predisposition from the approved intravenous Belimumab dose, that is 10 milligrams per kilogram. We could also see protective signals with the subcutaneous dose. Now, how to explain that? We all know that the high dose or the approved dose of intravenous Belimumab is now a drug that we use to treat active lupus nephritis. So the message could be confusing. However, we have seen, for example, when we looked at the cytokine profile of patients treated with Belimumab in Sweden, we saw a very quick and prominent decrease of IL-10. This was even more prominent and quicker than IL-6. we speculate that Belimumab, based on the dose, could have differential effects on different subsets of B cells, maybe B cells that have acquired regulatory properties, maybe during an active phase of the disease, are more affected by the high dose of Belimumab. The low dose of Belimumab maybe do not affect these B cell populations the same way. Yeah, so this is how I would summarize the implications. Then we shouldn't forget that in the BLISS-LN trial, for example, that resulted in the approval of the high dose, or the 10 milligram per kilogram dose of intravenous Belimumab, these patients had high-grade proteinuria. So one could expect in these patients a high drug clearance in urinary losses. So maybe the actual biologically active dose of Belimumab is not as high when the disease is active and gives a lot of proteinuria. So, but these are all speculations. It is, of course, important to really perform experiments and to look at the differential effects of different doses of Belimumab on B cells. Yeah.
Brilliant. Yeah, thank you so much, Iaonnis, for a great summary and discussion about your work. So I appreciate your time. So thank you everyone for listening. So you can follow me through my Twitter handle Yusof, and follow RheumNow either through Twitter, YouTube, LinkedIn for more content of EULAR coverage. Bye bye everyone.